for today's seminar. I first have to apologize or whatever to say. I'm just coming back yesterday from 2 weeks lecturing in Asia, Singapore, China, Taiwan. And unfortunately in Taiwan, my computer was stolen. I'm still waiting, hopefully getting it back. So I had to purchase a new computer and create new slides. Many people supported me in this way. So some of the slides that I originally had in the presentation will miss out, but I think it will be still a very interesting talk today. After 1 hour, about 1 hour, we will have 10 minutes break, then continue, and in the end, I think we have about half an hour for discussions. Please, when questions rise, put it in the notes. We will discuss them as good as possible in the end or maybe after the break. Yeah, well, as you see in this opening slide, there are many different forms of exosomes available these days. I could add another 100 bottles if I would like to cover all the different exosomes. I don't go into detail that every cell in our body, every cell in any human being, every cell in animals, every cell in the plants is secreting exosomes. I don't go into plant-based exosomes. I don't go into allergenic exosomes, so exosomes from goat, from sheep, or whatsoever, because I truly believe the exosomes from the human body are the best exosomes for treating humans. The exosomes are the same as their parental cells from which they are derived from. So a plant has so many different growth factors, cytokines, that are not necessary for us humans. Therefore, I don't go into plant-based exosomes and topics like this. However, we will discuss not only the most common use form of exosomes, the umbilical cord-derived exosomes, but we will go into the benefits of the huge range of other exosomes that are available these days. Second biggest topic today will be the NK cell, the natural killer cell-derived exosomes. We will go on the nervous cells-derived exosomes. We will go on to dendritic cell-derived exosomes. And we will go on a combination of NK cell, placenta-derived, and dendritic cells-derived exosomes. Few sentences to myself. I'm working in functional medicine for 3 decades. So I am in love with the mitochondria. Some 2 decades ago, more than 2 decades ago, I came into contact with photobiomodulation, the use of light for treatment of diseases, the combination of photosensitizers and light for photodynamic therapy in severe immune situations. I had many great results with functional medicine together with photomedicine. 6.5 years ago, we started to work with this tiny little extracellular vehicles. And since then, this became my next beloved topic. So I'm happy to share with you 3 decades functional medicine, more than 2 decades photomedicine, and more than 5 years use of exosomes, and in bracket, stem cells as well. Well, exosomes, one man's trash is the other man's treasure. I started my medical career in the 80s, and this was the time where research just found out about exosomes. And we learned it's trash. As the car has their exhausters. The cells need to wash out all the rubbish that may occur within the physiological processes, within the metabolic processes. And nowadays, we know it's the opposite. It's the most treasurous thing that one cell can give to another cell. Exosomes are the source for intracellular communication over big distances. Laser-like impulses, low-light impulses are very important for close cell-to-cell communication within the tissue, whereas the exosomes are responsible for the distant cell-to-cell communication. Already more than 20 years ago, it was observed that every single cell in our body secretes exosomes. And as the cell wall of the exosomes is almost exactly the same as the cell wall of the cells itself, they get easily absorbed, let's say digested by absorbing cells. So the exosomes are fantastic for delivering in the natural way proteins, cytokines from a distant cell to another cell. Nowadays, we may upload specifically exosomes for treatment purposes. So, not topic for today, but Western medicine realizes more and more the power of the exosomes as well. They, for example, try to load up exosomes with chemotherapeutic substances like cisplatin. And by this, they very easily, very specifically can deliver anti-cancer substances like cisplatin into cancer cells. We will see in a few minutes the basic information about the exosomes. The exosomes have roughly the size of hundreds to thousands of the cell. So therefore, the exosomes, and this is the first very important information, the exosomes easily penetrate from one tissue into the other. I'm using stem cells, but in many situations, I think the exosomes are much superior to the stem cells because they are so ultra small and they easily penetrate. We just mentioned the tumor blood barrier and bring their information into the tumor cells. They much more easily penetrate through the brain-blood barrier. So in neurodegenerative diseases, and I'm happy from some colleagues of the audience to share some fantastic clinical results by the use of the exosomes. So the exosomes are a unique form for cell-to-cell communication in the healthy body, and we can very specifically use exosomes derived from the different origins to support healing processes in the body. So they really can be not only a single treasure, but like you see here in the cartoon, they can be effective for a huge variety of different diseases. What are exosomes? How do they work? And why do they matter? As I said, they are tiny extracellular vesicles, and it's in reality quite often difficult. Is it an exosome or it's an extracellular vehicle? So in some conferences, they say, we shouldn't talk about exosomes. We should talk about extracellular vehicles because it's in the clinical situation, in the human body, very different. Is it a small 30, usually 80 to 120 to 150 nanometers, or is it a little bigger? Well, anyhow, extracellular vehicles and the exosomes are part of this covering group of extracellular vesicles. They carry proteins, lipids, RNA, microRNA, mRNA, and other signaling molecules. We will go into quite detail in this in a few minutes because this explains the powerful of the exosomes and why I believe that the exosomes are on average so much superior than conditioned media or so much superior than the use of proteins on its own. There are good indications, I don't want to argue on this, for the targeted use of proteins, but in the exosomes, especially in the umbilical cord-derived exosomes, we have a fantastic balance between pro-inflammatory and anti-inflammatory substances, so are very, very safe in most clinical indications. Well, the exosomes, the small vesicles, as mentioned before, enable cell-to-cell communication, especially between distant tissues. Key functions are the transfer of biological information between the cells. They support tissue repair and regeneration, I must say, on an almost unique form. They modulate inflammation and immune responses. We will go in the upcoming minutes on the different growth factors, vascular endothelial growth factors, hepatocyte growth factor, IGF-1, tumor growth factor beta, and so forth. We will look in which exosomes are which cytokines, and by this we already get a clear information about the best indication of which exosomes in which clinical situation. Modulate inflammation and immune response, influence skin regeneration and healing processes. I just wonder, when I use the arrow here, is it visible? Kann man den Pfeil sehen?
Yeah.
Yes,
good,
it's really well.
Good. Okay. Why are they important? They have fantastic clinical results today already in regenerative medicine and in facial regeneration, in aesthetic medicine. They definitely enhance the effect of the laser, or let's put it the other way around as well. The laser, very powerful, enhances the effectiveness of the exosomes. So if you look into the publications, started 40 years ago as the trash, and now as the treasure, brought into research more and more in a huge increase of different studies. So how exosomes react in the body and what are their effects? As mentioned before, tiny vesicles, natural messengers between the cells. They deliver bioactive molecules that support and guide, not only support, guide repair mechanisms, regeneration, and balance. Let's look into the 5 steps. Exosomes are primarily endosomes. They are within the cell and they get released from any tissue. They get released in a huge amount by which cells? Yes, by the stem cells. Stem cells are a powerful source of exosomes, but any tissue releases exosomes. And in brackets, for old guys like me, in the late 80s, we started with a PRP, with a platelet-rich plasma, which is, let's say, the grandfather or grandfather of the exosomes. However, some of the effectiveness of the PRP, the platelet-rich plasma, can be explained by the measured we produce the platelet-rich plasma, we produce a good amount of exosomes as well. Exosomes are so much more powerful than the PRP. I can assure you from long-term clinical experience. So exosomes are released by different cells. They travel through all the body fluids, not only through the bloodstreams, and get attracted, especially by injured cells, by damaged cells. They interact with the targeted cells and bind by specific receptors. And as mentioned before, having the same cellular wall, the same bilipid layer, they easily get digested by the receptor cells and bring all their information into the cells. So they work as fantastic cargo for proteins, mRNA, microRNA, lipid, growth factors, proteins. And here you see the inoculation of the exosomes. So they are good for cell regeneration and other processes. Let's go like this. Oh, one back. In summary, they act as inverted commas, almost intelligent messengers that communicate with cells and trigger a powerful biological response for regeneration, protection, and regeneration. Yeah, in the top of this slide, you see European Laser Academy. For 25 years, I'm working with especially low power laser devices, cold laser devices, and the first fantastic research that made me convinced about the power of laser light was the research showing, especially by Tina Carew, that laser targets mitochondria. I'm convinced that all chronic diseases are associated with injured mitochondria, with a depletion of mitochondria, with a reduction of healthy, and with a reduction of the number of mitochondria. And the laser targets these powerhouse cells, the powerhouse suborganal cells of our body target the mitochondria. And supporting these with the exosomes can be very, very powerful. So what is known about photobiomodulation? I could talk, for those who know me for a longer time, I could talk for weeks on the power of laser. So just 2 or 3 slides for photobiomodulation. But why is it fantastic to combine? Because in many things, they are not only additive, but boosting the effectiveness of each other. As said before, the main target for especially red and infrared light are the mitochondria. If we would have an attending seminar, I would now ask, now let's go to cellular physiology. What is the main target for red and infrared light irradiation? It's the complex 4 of the respiratory chain. It's the cytochrome C oxidase. By this is boosters. It supports, it outranges ATP production. So I may have a petri dish with yeast cells, put a laser shower on it, within half an hour, 150%. Half an hour, 150% more production of ATP. In the end, I will mention together with University in Sigmaringen, Professor Bergermann, Katja Matz, and Dr. Konzelmann, we are working on laser and mitochondria, and we are working on mitochondrial function, healing functions, and exosomes as well, different forms of our exosomes. It modulates oxidative stress. It supports angiogenetic signaling. And there we are already directly overlapping. What do the exosomes do, especially the umbilical cord-derived exosomes? I mentioned there are different growth factors, and the vascular endothelic growth factor, VEGF, is activated by the power of light and is one of the most important growth factors within the umbilical cord-derived exosomes. So they shake hands at this point. It helps to regulate inflammation. Laser enhances leukotaxis, so white blood cells go much more rapidly into the area where we irradiate. It boosts the activity of the phagocytes, so the digested coronaviruses and whatsoever get easier detoxified. And many of the growth factors we know from the exosomes do similar. Improve cellular metabolism. Well, I would say to improve cellular metabolism, there is nothing better than the laser with the best wavelengths, with the right color. Nothing else like this. As I mentioned about the ATP production, 150 to 400% increase within half an hour. Just mind-blowing, enhancing cellular membrane potential. So for cellular metabolism, the laser is unique, I would say. Enhances tissue repair pathways. Yes, definitely. Different growth factors are stimulated by laser irradiation. And I forgot to mention, if I have a tissue, irradiate with a laser shower or whatsoever, if I irradiate with class 3B laser from outside, what happens in the tissue? There is a significantly increase of release of extracellular vehicles and exosomes. So once again, these two love to shake hands and to collaborate. Enhanced tissue repairs. Hepatocyte growth factor, HGF, very important protein to regulate wound healings. So it's mainly expressed by our liver, but it's expressed by the other cells as well. So many overlapping things. Mitochondrial activation, ATP, energy boosting, and tissue priming, and to guide the exosomes. Let's say I have a patient with chronic inflammatory bowel disease, Crohn's disease, ulcerative colitis. When I give these, I would say probably at the best, the NK cell-derived exosomes, natural killer cells-derived exosomes, and use the laser shower or another tool over the abdomen, I will guide the exosome specifically into this area. And another thing, we know the exosomes have a very specific absorption spectrum. So azure light is the main absorption spectrum for the exosomes. So what we do in chronic diseases, we give systemic chronic diseases, we give infusions with the exosomes, whatever form, and then we do intravenous laser irradiation, for example, with the PhotoCare, the Moonlight Starlight device you see in the top of the cartoon. These offers all different wavelengths, from violet over blue, over green, orange, yellow, red, infrared. So we then very, very targetedly support the exosomes and support the other healing processes. Exosome response. Improve of cellular uptake potential. Use the right wavelengths. So main absorption pigment for most of the exosomes is azure, that means blue-green light irradiation. Better regenerative signaling environment. So I guide the exosomes where I want to get them. Enhance communication between damaged and repair cells. Well, when I have a better blood flow, the laser enhances micro and macro circulation. So it's a fantastic guiding tool to bring the exosomes into the desired area. Support for fibroblast, endothelial cells, immune cells, and enrichment of stem cells. It's another reason I mentioned, why do I love the exosomes more than the stem cells? All of them have their indications. Because if you do, for example, an infusion with stem cells, where do they go? In the first pass metabolism, they go, studies are a bit different, 70%, 80%, 82% into the lungs. So if I have a patient with COPD or chronic inflammation after COVID or whatsoever, I may be happy that most of the stem cells are harvested in the lung bed. But when I talk about patients with Parkinson, Alzheimer's, other neurodegenerative diseases, the absorption of the exosomes in the lung bed is so much less in comparison. And once again, the blood-brain barrier is so much easier penetrated by the exosomes than in comparison with the stem cells. Why combine exosomes with photobiomodulation? As mentioned before, they shake hands. It's a synergistic regenerative strategy. It's a synergistic anti-inflammatory strategy. Photobiomodulation, as mentioned before, uses mainly red and near-infrared light traditionally, because these were the first laser diodes that were available on the market. Nowadays, high-end devices like the Moonlight Starlight offers all different wavelengths for the targeted use of light. They help in the signaling of cell-to-cell communication. They help for on cellular level, on tissue level, on organ level, and if you do it intravenously, on the whole body level as well. We work in photobiomodulation with a non-thermal source of light. Some situations you may go for the much cheaper LEDs, the light-emitting diode, but for best information giving to the tissue for better penetration, the laser is much superior to the use of LED. All colors of the rainbow can be used for treatments. If you use from transdermal, it's only red and infrared. In most situations, we prefer the precise delivery of photons into the desired area. So I will share in a few minutes some slides where we treat omarthrosis or gonarthrosis. You could irradiate with red infrared from outside, but we want to bring the exosomes into the tissue. So why shouldn't we use the same needle by which we bring the exosomes into the degenerated joint, into the inflamed joint? Then we put a single-use sterile optic fiber after having injected the exosomes. We use blue light anti-inflammatory. We may use azure light to booster the exosomes. We use red light to enhance the ATP production and the repair mechanisms. If it's a bone tissue, we may add green light. Green light guides the development of osteocytes. Why it matters with the exosomes? Exosomes deliver on their own biological signals. The photobiomodulation prepares the tissue and guides in additionally the repair mechanisms. Together, they do support better cellular response. They do support better regenerative processes. Photobiomodulation improves microcirculation, inflammation, and guides tissue repair signaling. Together with colleagues from all over the world, we published some mind-blowing studies. At that time, 12 years ago, we didn't have therapeutic use of exosomes. Only by the power of light, we already could preserve so many foot and extremities in diabetic ulcers, chronic inflammatory wounds, by the power of light, maybe in combination with the photodynamic therapy. Exosomes may provide molecular cargo such as microRNA, proteins, and growth factors. And into these fields, we will go in a minute when we go into the different forms of exosomes that we can use. So, shake hand, synergistic effect. The last slide towards the laser or combination from molecular signaling to tissue level regeneration. Combined regenerative pathways, combined anti-inflammatory pathways. Photobiomodulation enhances mitochondrial function and energy production. We mentioned this. So what organs do need mitochondrial activation? What organs do have mitochondria for ATP production?
Is it a
Well, I
question?
think you all know. Pardon?
Is it a question? Shall they reply?
Well, I think unfortunately, it's a little bit difficult. How many are online now? 60. Thank you. Great. I guess all of you know. Every single cell, it was more a rhetoric question. All of you know every single cell in our body. The follicle of the hair roots, the sinusitis, mucous membranes, the thyroid gland, the brain, the heart, every single cell contains mitochondria. Without mitochondria, there is a very insufficient ATP production only. Without healthy mitochondria, we can only basic do anaerobic glycolysis. So from 1 unit of glucose, we only can produce 2 ATP. With a healthy mitochondria, out of 1 unit of glucose, we make 38 ATP. Wow, so much power, so much more powerful. So every cell contains mitochondria. One little exception, the grown-up red blood cells, the erythrocytes, pushed out the mitochondria. We have billions of mitochondria in the bloodstream as single mitochondria. We are still not in discussion what's their function in the bloodstream. The higher the energy amount of a cell is, the higher is the content of the mitochondria. So a healthy heart has 38%, 33 to 38% of the volume of a healthy heart are mitochondria. Wow. The mitochondria outrange the DNA in their genetic information and in their size. ATP support is the main tool of the mitochondria, and as the mitochondria are the main tool for photobiomodulation, they support the ATP production. They reduce the inflammatory burden, so it already starts to modulate inflammatory mediators. They have an influence on reactive oxygen species, on oxidative stress, and they bring the tissue already on a more acceptable level They improve the tissue readiness. They improve the environment for regeneration, healing processes. They improve the microenvironment for the use of the exosomes. If we go to the other part of the cartoon, the exosomes, they are biological signal carriers on their own. The laser activates substances that are responsible for signaling. The exosomes contain. So maybe for those who love, like me, not only functional medicine, not only regenerative medicine, but love Chinese medicine as well, they are a little bit like yin and yang. Laser, yang energy, powerful energy, exosomes, more substantial yin energy. So they shake hands. They are responsible for RNA, microRNA, messenger RNA delivery. They regulate genes of expression and cellular behavior, the way how often a cell replicates. They activate the resting stem cells. The laser does the same. If I irradiate a tissue with the right amount of light, as mentioned before, I activate the release of extracellular vesicles and exosomes, and I reactivate the sleeping stem cells. Both laser light as well as exosomes unlock the stem cells. Oh, I can assure you a wow effect. Growth factor signaling by exosomes stimulate proliferation, angiogenesis, survival of the cell. Laser as well. Laser has an influence that a cell doesn't go into the unprogrammed cell, does into necrotic, and similar things we know from the exosomes. Immune modulation balances the immune response, reduces chronic inflammation, and has a fantastic effect on matrix repair and remodeling of tissues. So when we go into the center, we see the synergistic regenerative effect of PBM, photobiomodulation, and the exosomes. We enhance cellular response and we achieve a significant superior repair and functional recovery. From all the people that have used the grandfather of the exosomes, the PRP, they said, yeah, we used it, we got some good results, especially in the younger people, in the sportsmen with partial rupture of the ligaments, Achilles tendon or whatsoever. But in the more elder patient, it was so and so. And I already told, listen, combine the PRP with the laser. I said, wow, it changed all our approach. And so much more powerful than the platelet-rich plasma are the exosomes. Yeah, the most common use, if you go into Google and say exosomes, you will find a lot of information about plant-based exosomes for facial regeneration. Well, once again, I don't think it's ideal. We are not plants. The plants protect themselves against too much UV irradiation. We are not plants. We have totally different structural proteins. What can we use with many of the structural proteins of the plants? At the best, they don't damage. At the worst, they don't guide any healing processes. So umbilical cord derived exosomes, from the umbilical, where do we have a huge amount of stem cells? Well, in our umbilical cord. So umbilical cord derived exosomes are a fantastic source for harvesting stem cells and for harvesting exosomes. The big, huge potential advantage, the big, huge advantage of exosomes is we are not talking about cellular therapies. We work with a cell-free regenerative therapy. I already mentioned in the very beginning, because of their small size, roughly a thousandth of the size of the cell, they much easier penetrate into the tissues. But at this point, I'd like to focus on the fact, if I work with not even a subcellular component, if I work with this ultra small B-layer vesicle, I have an extraordinarily low risk of adverse effects. However, I may mention at this point, and I think we have to keep this in mind, though, and I would claim it for all producers of exosomes, some are neglected, but for all producers in general, they try to get the best possible exosomes for the treatment. However, there are proteins in the exosomes. So if you have a patient who already had admission to intensive care unit with severe autoimmune reaction, with severe allergic reactions, we have to keep it in mind. I treated patients with severe multiple atopic conditions, but you have to know how to use. And maybe we have to have an additional seminar, a more practical, where we will record 1 or 2 treatments to see how many exosomes. I will mention, of course, how many exosomes, how many million exosomes or billion exosomes for which conditions, how to dilute it. But this is another more practical thing. So we have a cell-free regenerative therapy with a very low risk of adverse effects. Low immunogenicity. Supports tissue repair and wound healing, as we already mentioned several times. And I will bother you in this when we look into the different growth factors later on as well. Promotes vascular regeneration. I already mentioned the most prominent, the most detailed research, the VEGF, the vascular endothelial growth factor. This is probably the most well-described factor by the umbilical cord-derived exosomes. It may reduce excessive inflammation. In brackets, when I have excessive inflammation, then I would go more for our natural system again. Just think physiological. And this is, in brackets, my big argument with what we call Western medicine. We have so fantastic research. We have so fantastic insight into physiology and pathophysiology. But quite often, we look narrow-minded onto a disease. We see a symptom, jump on the symptom, and forget totally about the physiology and pathophysiology behind it. So, looking on severe, massive inflammation, any degenerative process is inflammation, it's an intoxication with ROS, with reactive oxygen species. And if it's significant, we have to think, are the umbilical cord derived the best, or shall we go for NK cell derived exosomes? Excellent safety profile in experimental studies, together with different institutes, not only with the University in Sigmaringen in Germany, but together with different institutes in India, we have conducted some very, very deep safety studies, and generally the umbilical cord-derived exosomes can be regarded as very, very safe methods for treatment. Yeah, they are suitable for advanced protocols in regenerative medicine. If we look into the publications, there is a huge variety for the use of the umbilical cord-derived exosomes in regenerative medicine in general, in reducing pro-aging processes, mind-blowing in studies on wound healing, in orthopedics. I will show you some pictures on the different osteoarthritis situations, dermatology and aesthetics medicine, hair restoration. Once again, the overlapping. Laser light, infrared laser can reactivate the sleeping, the dormant follicle cells for the hair growth. So most research shows in many conditions, it's not that the follicles are dead, but they go into a sleeping situation. And just fantastic in this is injection of umbilical cord-derived exosomes. We do it centimeter by centimeter by centimeter by centimeter, and then use photobiomodulation to enhance the circulation, to enhance the lymphatic drainage, to booster the ATP production. Tissue regeneration, we have a huge number of so-called growth factors, of proteins that are necessary, are required for tissue regeneration. Chronic ulcer, great results in patients even with poor controlled diabetic ulcers. However, doing functional medicine, we have to keep everything in mind. First of all, of course, we have to normalize the HbA1c, the hypoglycemia, no doubt about it. But in all these patients, almost all, we see an outrage of inflammation. We see high levels of so-called lipid peroxidation. We see high levels of the oxidized LDL cholesterol. It's not the cholesterol in general it's dangerous. We need cholesterol to survive, to produce many of our hormones. We always say we have the good, the HDL cholesterol, and the dangerous LDL cholesterol. But the LDL cholesterol only gets then dangerous when it's oxidized, when it's under oxidative stress. And we nowadays can measure the oxidized LDL cholesterol. Musculoskeletal regeneration, well, the sports people love the exosomes, and yeah, I have treated football players from the first primary league in Germany, from the Bundesliga, as well as a tennis player, 60 years, 64 years of age, who still wanted to play tennis. And due to recurrent ankylosing and issues with the menisci and both knees, he was not able to play anymore. He went to playing golf, but was not happy with this. He's playing on a regular basis, 2 to 3 times a week again, his beloved tennis. Yeah, this is probably, let me look on the time. Yeah, almost 1 hour already. This is probably my most beloved slide from the presentation. It's going a little bit deep into regulatory processes, but it explains really in detail how do exosomes work and why are they so absolute mind-blowing, fantastic in regenerative medicine. Cell-free regeneration, signaling, tissue repair, healing. We heard these terms so often. Umbilical cord-derived exosomes are extracellular vesicles secreted by cells from the human umbilical cord, particularly from the stroma cells. Therefore, quite often you see UC, MSC, umbilical cord, mesenchymal stem cells derived exosomes. They transport, and this is the key thing, they transport a diverse, huge range of different cargo of proteins, lipids, and regulatory RNAs that participate in intracellular communication and support regenerative biological processes. So we have a huge number of different growth factors in the umbilical cord-derived exosomes. And the first here in the list, I may go just here, is what I already mentioned several times before, the vascular endothelial growth factor. It's not one growth factors, but if we go into the research, into the papers, we see it's a group of signaling proteins, and as given by the name, or as you know, one of the main growth factors or targets is the angiogenesis. So by applying the vascular endothelial growth factors, we especially booster the neoangiogenesis. And this is exactly what we know from laser irradiation. So another shake hand. Then we have, I mentioned briefly already as well, the HGF, the hepatocyte growth factor. It's a protein that controls cell growth, and not only the cell growth, but controls cell movements as well, and guides tissue repair. So quite often, we only think about one function, but many of these growth factors and cytokines have a variety of different effects, and sometimes a real balancing effect between pro-inflammatory and anti-inflammatory effect. So hepatocyte growth factors, especially as the name gives, produced, expressed by the liver. And we know the liver has fantastic repair mechanisms. It's one of the organs with the most powerful self-healing capacities, cell growth, cell movement, tissue repair. It makes cell scatter and move through wounds. In wound healing, one of the most important substances. And as it scatters the substances, sometimes it's not called the HGF, but it's called scatter factor. And like the vascular endothelial growth factors, another effect between cell growth, movement, it's the effect on angiogenesis. We have the IGF-1, insulin-like growth factor, insulin-like growth factor number 1. It's a very important hormone in many parts of our body. It is like the HGF-F, mainly synthesized in the liver, and it is one of the most important hormones for muscular regeneration and bone regeneration. So I mentioned in the slide before, what are the indications for umbilical cord-derived exosomes, especially osteoarthritis, muscular impairment, muscular ruptures. Umbilical cord-derived exosomes, by especially the insulin growth factor, are in general, the exosomes of choice in this situation. Under the control of or the IGF-1 is under the control. Everything in the body is nicely communicating. The insulin-like growth factor is under the control of the growth factor, of the growth hormone, let's say, of the growth hormone that is released by our pituitary gland. So there's lots of communication and regulation processes in the body. There is a feedback metabolic process between the production and release of the insulin growth factor and the pituitary gland. Insulin-like growth factor, what does it mean? It has some mimicking effects to insulin, otherwise it wouldn't be called like this. And like insulin, what is insulin? Insulin has an anabolic effect, so the insulin-like growth factor promotes protein synthesis and has an anabolic effect as well. Then we have the TGF-beta, transforming growth factor beta. It's a cytokine that is important for growth and proliferation, for cell differentiation, and for the immune system. So again, I'd like to stress, usually we keep one thing in mind. Vitamin D, bone. Vitamin D, calcification of the bone. Oh, no, vitamin D is it's not a real vitamin, it's a hormone. It has so many effects for the body. It's probably the most important vitamin for the immune system. It presents the antigen for the immune system. So the TGF-beta is not only for cellular growth, but proliferation, differentiation, and an important factor for the immune system. And it balance growth and proliferation. At the same time, it's very powerful in its anti-inflammatory effect. Last here on the first column is the FGF, the fibroblast growth factor. Once again, it's not one growth factor, it's a huge variety of different signaling proteins. The most common among this fibroblast growth factor is so-called factor 23, a central hormone in the human body, and where it's produced mainly in the osteocytes. So why I believe umbilical cord-derived exosomes, osteoarthritis, for example, because here we found a good number of the fibroblast growth factors. Last thing I'd like to go into in more detail is the first on the second of the column is the IL-10, the interleukin 10, one of the most powerful anti-inflammatory interleukins. Very pro-inflammatory is IL-6, very anti-inflammatory, powerful anti-inflammatory cytokine is the IL-10. It's a central role in regulation of immune responses. It suppresses, it balances, it brings down the pro-inflammatory cytokines, such as what is the most common used, most common described pro-inflammatory cytokine beside IL-6. It's the tumor necrosis factor alpha. It reduces and balance IL-6, IL-1, and the tumor necrosis factor alpha. By this, I would say prevent, but it reduces the destruction of tissues. And why I said before, why do we forget why we have it on a gold plate? We get it presented by physiology and pathophysiology. Why the hell physiology in the universities is a hated topic? When we talk about, for example, inflammatory bowel diseases, when we talk about ulcerative colitis, when we talk about more Crohn's disease, we know all these patients have a significant low level of IL-10. We have a tool how to treat them, and still we go for corticosteroids, anti-inflammatory, and we don't care about all the side effects. We have so many things in our hand that we could use in a natural way in many situations. And last thing about IL-10, what is the worst inflammation that we can? The septic shock. And once again, IL-10 is a very powerful shield against the septic shock. Yeah, looking at all this, you may understand my love for exosomes and my love for umbilical cord-derived exosomes in the right indication. Modes of action, modulation of inflammatory processes. We went into some details into the different cytokines and growth factors, promotes angiogenesis, stimulates fibroblast proliferation, enhancement of collagen synthesis, and extracellular matrix remodeling. Hey, what's the effect of laser light? Laser light activates fibroblasts, activates elastin, proline production in the tissues. So if I go for injection of exosomes, I strongly would shake hand and advise to use a laser shower into the areas where I worked with the laser. Support of stem cell mediated tissue regeneration. I mentioned before, exosomes unlock local stem cells. All our tissues are full of stem cells. When we talk about stem cell transplantation 20 years ago, we only thought about stem cells derived from the bone marrow. I did it in my clinical times at the university, go into the tibia, tuberosus of the tibia bone or the anterior posterior iliac spine to harvest stem cells and give in patients with severe immune depression. With the laser, we activate stem cells and extracellular vehicles, and with the exosomes, we unlock the power of the stem cells as well. Yeah, I think though we are already over the hour that I aimed for, here a patient a few days ago, primer football player, successful football player, where we injected umbilical cord derived exosomes. You may combine it with hyaluronic acid. I strongly would suggest not to combine it with corticosteroids. I strongly would suggest, on the other hand, to combine it with photobiomodulation. Those who know my approach, who have seen lectures from me before, we go under strict sterile conditions into the joint space directly. Of course, sterile needle. By this, we inject the exosomes, and then we put a sterile single-use optic fiber in. We always start with the blue light because blue light is the most powerful anti-inflammatory substance. With blue light, we can kill viruses, bacteria, fungi, reduce inflammatory processes in general. Then we go usually with the red light or red plus green light irradiation. Some people harvest stem cells from abdominal fat. I mentioned the bone marrow. We have so much more stem cells in the abdominal fat, but what is in the abdominal fat as well? Huge number of stem cells, yes. 10 times, 100 times more per ml than in the bone marrow. And even younger stem cells with a higher differentiation potential. But what is abdominal fat known for? Of course, you all know. It's one of the main sources for inflammatory process. When we talk about chronic inflammation, autoimmune diseases, where do we see it? Quite often in the very obese patient with huge amount of abdominal fat, and there is a huge amount of inflammation. And if I harvest stem cells and don't grow them in the lab, but just harvest them and produce them in my own lab without growing them, without expanding them and inject them, I inject a huge amount of pro-inflammatory macrophages. This is the reason why people who do this combine with corticosteroids. Big no-no. I wouldn't advise to do so. And if you go to the conferences for regenerative medicine, they usually say, no, no, no. The stroma vascular function is not stem cells. Of course, some stem cells, but more fat cells and many huge amount of macrophages. So I can assure you the photobiomodulation is so much more effective combined with the right exosomes. Here, a patient who came all the way from India for treatment, umbilical cord-derived exosomes plus blue and red light irradiation. We injected, I'll give you one clinical first, very clinical guideline on each side, 40 billion of exosomes. And I can assure you there are few products with such a high amount of exosomes, and you never should inject 40 billion in the face in a single place. And we followed up with blue and red light irradiation each for 10 minutes. And here is a graph of one of the devices we use, the photon care device, where you see we have a huge variety from the very short wavelengths of violet or blue, green, yellow, red, and infrared for